Hi everyone, welcome back to this video lecture series. This lecture covers the cell cycle and mitosis. In this lecture, we will cover the roles of cell division, chromosomes and chromatids, somatic cells and gametes, the phases of the cell cycle, mitosis and interphase, binary fission, and the checkpoints of the cell cycle, which could result in cancer with loss of regulation. After this lecture, students will be able to describe the major functions and events of cell division that enable the genome of one cell to be passed on to two daughter cells. In particular, you'll be able to differentiate between mitosis and binary fission, list the phases of the cell cycle and describe the sequence of events that occur, and explain how the abnormal cell division of cancerous cells escapes normal cell controls. Before we get into the specifics of cell division, let's discuss the key roles of cell division. Living organisms have the unique ability to produce more of their own kind, and this distinguishes them from non-living matter. Plant cells, animal cells, bacterial cells, fungi cells, and all other cells have this ability. The continuity of life is based on this unique ability to reproduce cells, and we call this cell division. In unicellular organisms such as bacteria and amoebas, division of one cell is what reproduces an entire organism. In multicellular organisms, thousands to trillions of cell divisions occur within the span of a life. For humans, there are approximately 3 times 10 to the 13th cells in the body, and that's 30 trillion cells, and 10 to the 16th cell divisions in an entire lifetime, which is 10 quadrillion cell divisions. Keep in mind, this is a total number of cell divisions. Each individual cell can only divide between 50 and 70 times before it dies. And remember, we all begin as just one cell, a merging of sperm and an egg, and we'll talk about this in the following chapters. Multicellular organisms depend on cell division for 3 things: development from that fertilized cell, growth, and repair. Cell division is an integral part of the cell cycle, which encompasses the life of a cell from its formation to its own division. You should be familiar with the term genome. All of the DNA in a cell constitutes the cell's genome. A genome can consist of a single DNA molecule, often a circular piece of DNA, which we call a plasmid, and we see this here. And this is common for prokaryotic cells such as bacteria and is located in the nucleoid region for them. You might also recall from lecture 6 that mitochondria and chloroplast also have their own circular DNA. Or a genome can consist of several double-stranded linear DNA molecules, which is common for eukaryotic cells. DNA molecules in a cell are packaged into structures, and we know that they are called chromosomes. And as we know, human somatic cells each contain 46 individual chromosomes, and they're of course located in the nucleus for eukaryotic organisms. This is an image of a karyotype, which shows all of the chromosomes for this individual organized by length. Before we get into the stages of cellular division, we have to take a closer look at chromosomes. Eukaryotic chromosomes consist of chromatin, which is the complex of DNA and histone proteins, which condenses during cell division. This is shown in the image on the right. Wrapping the DNA around those histones prevents the strands from becoming tangled, which prevents the DNA from damage, and we do not ever want to damage our DNA. Every eukaryotic species has a characteristic number of chromosomes in its nucleus. Again, for humans, we have 46 chromosomes. And to contrast this, we will be learning about a species called Drosophila. They're fruit flies, and they have only 8 chromosomes in their nucleus. In this course, we're going to be learning about the 2 types of cell division, and I'm sure you've heard of them before, mitosis and meiosis. Most cell division results in genetically identical daughter cells with identical copies of DNA, and this type of division is called mitosis. The other type of cell division, meiosis, is a very specialized type of division that produces our sex cells, sperm and egg. And we'll talk about this in depth in the next lecture. But for now, just make sure that you understand that mitosis produces 2 copies, they're identical daughter cells, while meiosis produces 4 non-identical cells. And we will cover each stage of mitosis in just a bit. So there are 2 main types of cells in our body, and I've just mentioned them already, but they are somatic cells and gamete cells. Somatic cells are produced by mitosis, and they are non-reproductive. In other words, somatic cells are all of the cells other than the sperm or the egg, which are our reproductive cells. This includes muscle cells, skin cells, blood cells, neurons, bone cells, liver cells, kidney cells, and all of the other cells within our body other than the sperm and egg. Somatic cells all have 2 sets of chromosomes. One set you inherit from your mother, the other from your father. And the other type of cells are the gametes. Gametes are our reproductive cells, which are the sperm and the egg. Gametes are special in that they have half as many chromosomes as the somatic cells do. And these special cells are produced by meiosis, which again yields non-identical daughter cells that have only 1 set of chromosomes, half as many as the parent cell. And again, we're going to cover how this happens and why in the next lecture. To prepare for cell division, the DNA has to first be replicated. And we are also going to be learning the details about DNA replication on the cellular level in lecture 16. But for now, just note that the DNA has to be replicated during the cell cycle before we go through cell division. After replication, the individual chromosomes condense. We'll use a human cell as an example. It begins with 46 chromosomes. DNA replication doubles that number to 92. And we say actually 92 chromatids. And that's a lot of strands of precious genetic material that the cell needs to keep track of. So the cell has a trick to keep the chromatids organized. And the 2 identical copies of DNA need to remain tethered together. And they're attached by something called the centromere. And this is what gives the chromosome that X shape. Anytime you see an X, that's actually 2 chromatids held together, and those chromatids are identical. They are sister chromatids. They're exact copies of each other. So what is a centromere? The centromere is a narrow waist of that duplicated chromosome where the 2 sister chromatids are most closely attached. These 2 completely identical molecules, again, we call them sister chromatids. And together, the 2 sister chromatids attached at the center by the centromere make up 1 chromosome. So 2 sister chromatids held at one centromere make up one chromosome. We actually count chromosomes by counting centromeres, just as a little trick. Notice the terminology that I'm using here. 2 sister chromatids, which are the identical strands of DNA condensed with the help of proteins and held together by the centromere, are considered one chromosome. These 2 sister chromatids are what separate during mitotic cell division to ensure that each daughter cell has one copy of each of our 46 chromatids. Just imagine if a cell got 2 copies of the same chromosome or chromatid. That would be bad. We don't want duplicate copies like that. This is why the centromere is so important, and this is why keeping sister chromatids together is so important. It's an easy way of the cell organizing the genetic material so that we separate it properly. Here is a basic model of replication and division. This cell begins with one chromosome consisting of one chromatid. We see on the right the 2 strands of chromosomal DNA molecules in a double helix for this one chromatid. In box 2, the chromosomes have replicated. To the right, we see 2 double helices consisting of 4 strands of chromosomal DNA. These 2 chromatids are identical, and these are called the sister chromatids. The sister chromatids are held together by the centromere, and during cell division, the 2 sister chromatids of each duplicated chromosome separate and move into 2 different nuclei. Once separate, the chromatids are called chromosomes. We have now created 2 identical daughter cells with identical genetic material. So now let's take a deeper look into the distinct phases of the cell cycle. The figure shown is a representation of an entire cycle of a cell. There are 2 main phases of the cell cycle, interphase and mitotic phase. First, let's mention mitotic or M phase. If you've ever taken a biology course, you've probably already learned the phases: Prophase, Prometaphase, Metaphase, Anaphase, Telophase, and Cytokinesis. And we're going to go into the details in just a moment. Next is interphase. Locate interphase on this figure. You'll notice that for most of the cell, about 90% of this cycle, the cell is in interphase. Interphase is the part of the cell's life involved with cell growth and replicating the DNA in preparation for cell division. It's also comprised of any and all other cellular processes that are allowing the cell to function and stay alive. We see that interphase is further divided into 3 phases: G1, S, and G2. G1 means first gap. During this phase, many proteins and nutrients are made in preparation for replicating that DNA. Here, the cell is accumulating the building blocks of DNA and the associated proteins, as well as accumulating whatever energy is necessary to complete the task of replicating the DNA. The next phase is the S phase. The S phase means synthesis, and this is because this is when the DNA is actually replicated or synthesized in the cell. The identical pairs of DNA molecules, called sister chromatids, are firmly attached at the centromere, and the centrosome is also duplicated during the S phase. We'll talk about the centrosome in just a moment. You might recognize that from the chapter on parts of the cell. The third phase is G2, which means second gap. In this phase, the cell replenishes its energy stores and synthesizes any other proteins that are necessary for chromosome movement. And here we're also duplicating some of the cell's organelles, and we're dismantling that cytoskeleton, because we can utilize it as resources for the mitotic phases. There may also be some additional growth of the actual cell during the G2 phase. By the end of interphase, the cell has grown, we've accumulated any necessary proteins and energy, we've replicated the DNA, and we're prepared for cell division. The next phase is the mitotic or M phase. Mitotic phase denotes the beginning of mitosis, which is the division of the genetic material in the nucleus. Note that mitosis is particularly the division of the genetic material, the DNA, in the nucleus. The process of mitosis is, of course, further divided into 5 phases, which you all know: Prophase, Prometaphase, Metaphase, Anaphase, and Telophase. Following telophase, there is a special process called cytokinesis, whereby the cytoplasm of the 2 newly created cells divides to separate into the 2 identical daughter cells. So note that cytokinesis has nothing to do with the genetic material. It's not technically a part of mitosis. It is a separate process specifically for where the cytoplasm of the cell divides. So we're going to walk you through the phases now. And remember, mitosis is the division of the genetic material, i.e. the DNA in the nucleus. There are a few major things to pay attention to at each phase. First is what shape the chromosomes are in. Are they condensed? Are they loosened and opened? Next is the mitotic spindle and the centrosomes. Where are the centrosomes, and is the spindle attached to the chromosomes yet? The spindle attaches to the kinetochores and the sister chromatids, and I'll explain that in a little bit more detail. And finally, the nuclear envelope. Is it formed? Is it broken? These are all the things we're going to be paying attention to as we progress through the phases. So prophase is the first official phase of mitosis. In prophase, we can see that the chromosomes have already begun to condense in the nucleus. Close observation will also show that each chromosome is composed of 2 sister chromatids and that they are attached at the centromere, which is why the chromosomes look like an X. The nuclear envelope is intact here, although it is beginning to break down, and the centrosomes have begun to move to opposite sides of the cell with a little bit of early mitotic spindle emanating from them. The nucleolus, which is inside the nucleus, also disappears in this phase. Next is phase 2, Prometaphase. The chromosomes are now completely condensed, and the sister chromatids are still attached together at the centromere. The nuclear envelope is further disintegrating, which is now allowing for the mitotic spindle to reach the sister chromatids. The centrosomes have also reached the opposite ends of the cell here. This is a good point to talk about the mitotic spindle and that special structure called the kinetochore. Take a look at the structure called the centrosome, which is shown here in yellow. Each cell begins with one centrosome, which is duplicated during interphase. They begin together on one side of the cell, but they migrate to opposite poles of the cell during mitosis. Centrosome is the term for that entire amorphous structure, but the organelle that's responsible for its actual function is called the centriole. And you can see the 2 pairs of centrioles that are pictured in the cell. Each centriole kind of looks like a rigatoni pasta. The role of the centriole will be formation of the mitotic spindle, which is the structure, you should recall, made of microtubules that controls chromosome movement during mitosis. In animal cells, the assembly of spindle microtubules begins in the centrosome, the microtubule organizing center. An aster, which is the radial array of short microtubules, extends from each chromosome. And an easy way to remember that is the root word for aster is star, which is why you might recognize it in astrology, astronomy. And the aster looks like light emanating as in from a star. The spindle includes the chromosomes, the spindle microtubules, and the asters. I think of the mitotic spindle kind of like Spider-Man's web. Spider-Man is the centrosome, and his mechanical web shooters are the centrioles. The purpose of the spindle is to grab onto the condensed chromosomes. Remember, there are 2 centrosomes, one at each end of the cell. So the spindle is being shot out from both ends of the cell. Once each chromosome has been grabbed onto by the spindle from both centrosomes, the sister chromatids can be pulled apart. Now, there is one major difference between how Spider-Man functions and how the mitotic spindle separates the sister chromatids. Spider-Man is physically pulling the web using force to move the objects that they're attached to, but this does not happen in the cell. In other words, the centrosomes are not pulling the chromosomes to them. Instead, the cell has a mechanism called a kinetochore to move the chromosomes apart. A kinetochore is a complex of proteins that is associated with the centromere that the mitotic spindle can reach out and attach to. Remember, kinetic energy has to do with motion, and the word kinetic is in kinetochore. So kinetochore is a structure that facilitates movement of the chromosomes. The microtubules, the mitotic spindle microtubules are not pulled again. Instead, the kinetochore is able to move itself up along the microtubules. And in doing so, the microtubules become shortened by depolymerization, which just means that the monomer subunits of microtubules, which is tubulin, gets chopped off as the kinetochore ends move up along the spindle. In prometaphase, some of the mitotic spindle reaches out from the centrioles to attach to these kinetochores, while other spindle microtubules attach to opposite strands of spindle microtubules. The spindle microtubules that are attached to the chromosomes begin to move them apart within the cell, but the other spindle microtubules, which do not attach to the chromosomes, are called non-kinetochore microtubules, and they will overlap and push against each other to help elongate the cell. The third phase of mitosis is metaphase, which I think is the easiest phase to recognize and remember. At metaphase, the chromosomes have been assembled at the center of the cell, which we call the metaphase plate, and this was due to the movement of the kinetochores. I want to bring to your attention a few important aspects of metaphase of mitosis. Remember, it's the sister chromatids which are lined up at the metaphase plate, and the sister chromatids are completely identical. We see the centrosomes at opposite ends of the cell with their asters of spindle microtubules emanating and attaching to the chromosomes. Spindle from both sides have attached to each chromosome centromere, and this is crucial because this is how the identical sister chromatids will be separated from each other. This is so important that it's actually a checkpoint for the cell. If the mitotic spindle is not attached on both sides, the cell will abort mitosis. We'll return to this at the end of this lecture. We'll talk a little bit more about checkpoints. The fourth phase of mitosis is anaphase. In anaphase, the replicated chromosomes are split, and the identical sister chromatids can move to opposite sides of the cell. How do they do this? They are able to move by the mechanism of the kinetochore. The kinetochore functions by moving along the microtubules toward the opposite ends of the cell. As it moves along the mitotic spindle, which is made of microtubules, the microtubules shorten by depolymerization. From chapter 4, we know what it means to make a polymer or to polymerize. So again, depolymerization is the deconstruction of a polymer, in this case, the microtubule chain, by removing monomers of tubulin protein as it moves. Those non-kinetochore microtubules from opposite poles are overlapping and they're pushing against each other to help elongate the cell. In the final stage of mitosis, telophase, genetically identical daughter nuclei begin to form at opposite ends of the cell. So here we see the reformation of the nuclear envelope to surround and protect our DNA. Each cell also contains one centrosome with its pair of centrioles. Telophase and even parts of anaphase can happen in unison with cytokinesis, which is the division of the cytoplasm. Again, mitosis is the division of the genetic material. Cytokinesis is division of the cytoplasm. You'll also notice the cell is beginning to pinch in the center. In animal cells, cytokinesis occurs by a process known as cleavage. The pinched in part of the cell is called the cleavage furrow. This is accomplished by a contractile ring of microfilaments that tighten and squeeze until the cell is pinched into two. Plant cells have a different process for their cytokinesis. Remember, plant cells have both a plasma membrane and that rigid cell wall, which is made of cellulose. During cytokinesis for plant cells, a cell plate forms to separate the two identical daughter nuclei into separate cells. I'm a big proponent of repetition for biology, so just to drive it home, let's recap the role of the mitotic spindle at each of the phases. So for prometaphase, some of the spindle microtubules have attached to the kinetochores of the chromosomes, and they begin to move those chromosomes toward the center of the cell. At metaphase, the chromosomes are all lined up at the metaphase plate, which is that imaginary line right at the midpoint between the spindle's two poles. During anaphase, the sister chromatids separate and move along the kinetochore microtubules toward opposite ends of the cell. In telophase, genetically identical daughter nuclei form at opposite ends of the cell, and cytokinesis begins during anaphase or telophase, and the spindle eventually disassembles. So there you have it, the phases of mitosis. We discussed when and why the chromosomes condense, formation of the mitotic spindle, centrioles and centrosomes, how and when the kinetochores move the sister chromatids apart, and when and where the nuclear envelope breaks. Always remember, mitosis is about the division of the genetic material, the DNA, and all things that are involved in that process. Prokaryotes like bacteria and archaea have a unique form of cell division called binary fission. In binary fission, the chromosomes replicate beginning at the origin of replication. And the origin of replication is just what it sounds like it would be. It's a special part of the genome that initiates DNA replication. Remember, prokaryotes like bacteria have a circular genome called a plasmid, unlike eukaryotes, we have multiple strands of linear chromosomes. Once the replication of the genome is complete, the two daughter chromosomes actively move apart and the plasma membrane pinches inward, dividing the cell into two. This is an image of an agar plate with some plated bacteria, and we can see that the bacteria plates in different colonies. So each of those dots is a colony of genetically identical bacteria. So they are all clones within each dot. The eukaryotic cell cycle is regulated by a molecular control system, the cell cycle control system. The frequency of cell division varies with the type of cell. For example, the cells in the skin and those that line your intestinal epithelium are the fastest dividing cells in the human body. These differences result from regulation at the molecular level. The sequential events of the cell cycle are directed by a distinct control system, which is very similar to a clock. As you see in the image, the clock has specific checkpoints where the cell cycle stops until a go-ahead signal is received. We have 3 distinct checkpoints here in red, so let's look at each of them. First, let's note that the cell cycle control system is regulated by both internal and external signals at these checkpoints. An example of an internal signal was given while describing the metaphase phase of mitosis. If you remember, if the kinetochores do not attach to the spindle microtubules properly, a molecular signal is sent out that delays anaphase. Some external signals are growth factors, which are proteins released by certain cells that stimulate other cells to divide. A clear example of an external signal is density-dependent inhibition, in which crowded cells stop dividing. Most animal cells also exhibit something called anchorage dependence, in which they must be attached to a substratum which acts as a foundation in order for them to divide. Cancer cells exhibit neither density-dependent inhibition nor anchorage dependence. For many cells, the G1 checkpoint seems to be the most important. If a cell receives the go-ahead signal at the G1 checkpoint, it will usually complete S, G2, and M phases and then divide. If the cell does not receive the go-ahead signal, it will exit the cell cycle and switch into a non-dividing state, which is called G0 phase. Cells that are in G0 phase are not actively preparing to divide. The cell is in a quiescent or inactive stage that occurs when cells exit the cell cycle. Some enter G0 temporarily due to environmental conditions such as the lack of availability of nutrients or stimulation from growth factors. The cell will remain in this phase until conditions improve or until an external signal triggers the cell to enter the G1 phase. Other cells that never or rarely divide, such as mature cardiac muscle cells and your nervous system cells, remain in G0 permanently. The G2 checkpoint bars entry into the mitotic phase if certain conditions are not met. As with the G1 checkpoint, cell size and protein reserves are assessed, and the most important role of the G2 checkpoint is to ensure that all of the chromosomes have been replicated and that the replicated DNA is not damaged. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted and the cell attempts to either complete its DNA replication or repairs the damaged DNA. The M checkpoint occurs near the end of the metaphase stage of mitosis, and we already discussed it a little bit before. The M checkpoint is also known as the spindle checkpoint because it determines whether all of the sister chromatids are correctly attached to the spindle microtubules. The cycle will not proceed until the kinetochores of each pair of sister chromatids are firmly anchored to at least 2 spindle fibers arising from opposite poles of the cell. This way, when anaphase commences, the 2 sister chromatids will be pulled apart and separated properly in different directions. 2 groups of proteins called cyclins and cyclin-dependent kinases, which is abbreviated as CDKs, are called positive regulators. And this is because they are responsible for the progress of the cell through these various checkpoints. Increases in the concentration of cyclin proteins can be triggered by both external and internal signals. Cyclins regulate the cell cycle only when they are tightly bound to CDKs. To be fully active, the CDK cyclin complex must also be phosphorylated. CDKs are enzymes. Remember, kinases are enzymes that phosphorylate, meaning they add a phosphate group onto other proteins, and phosphorylation activates the protein by changing its shape. The proteins phosphorylated by CDKs, cyclin-dependent kinases, are involved in advancing the cell to the next phase. So now you've learned the cell cycle. Interphase, which is comprised of G1, S, and G2, and mitotic M phase, which is comprised of prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Almost as important as understanding the phases of the cell cycle is understanding how the cell cycle is regulated in eukaryotic cells. Even without knowing it, you all know one consequence of irregular cell cycles, and this is cancer. There are, of course, many types of cancers and countless causes, but a notable cause of cancer is dysregulation of certain genes, which can lead to loss of cell cycle control. Here's 2 types of genes which could result in a cancer cell. Proto-Oncogenes are the genes that code for the positive cell cycle regulators, meaning they will make the cell cycle go. Proto-oncogenes are normal genes that, when mutated in certain ways, become oncogenes, which are genes that cause a cell to become cancerous. You probably recognize the part of the word for oncogene, onco. An oncologist is a doctor who specializes in treating cancer patients. Tumor suppressor genes are segments of DNA that code for negative regulator proteins. So these genes can prevent the cell from undergoing uncontrolled division. We see the example here is a gene for p53. If we have normal p53, this protein will be able to cause the cell to go through apoptosis, which is programmed cell death. So if there's anything wrong with this cell, this protein will help end the cell. And so that cell won't continue to divide and create more bad cells. However, if there's something wrong with this gene and it's mutated, and we create a p53 protein that doesn't work properly, it can't stop the cell from dividing. And so the cell cycle is going to continue, and we can have cells that are cancerous. Cancer cells do not respond normally to the body's control mechanisms. In fact, cancer cells may not even require growth factors from the body to grow and divide. This can happen for a number of reasons. They may be able to make their own growth factors. They may convey a growth factor's signal without the presence of the growth factor, or they may have an abnormal cell cycle whereby the checkpoints no longer serve to halt division of unhealthy cells. These factors may lead to the formation of a cancerous cell. When a normal cell is converted to a cancerous cell, this process is called transformation. Other terms for this are malignant transformation or tumorigenesis. Cancer cells that are not eliminated by our immune system can form tumors, which are masses of abnormal cells within otherwise normal tissue. If these abnormal cells remain only at the original site, the lump of cells is called a benign tumor. Malignant tumors are different. They invade surrounding tissues and can metastasize. Metastasis is the spread from a primary site to a secondary site. So metastasis exports cancer cells to other parts of the body where they may form additional tumors. In this image, we see a tumor present in a mammary gland tissue. This tumor began from a single cancer cell. As the cells in the tumor continue to divide due to the dysregulation of the cell cycle, the tumor cells invade the neighboring tissue. Eventually, the cancer cells spread through the lymph and blood vessels to other parts of the body. Recent advancements in understanding the cell cycle and cell cycle signaling have led to advances in cancer treatment. And that will wrap it up for this chapter. In the next chapter, we'll learn all about meiosis and we'll be able to compare it to mitosis.